Good day, everyone. Okay, continuing with our sessions for your in biology, strand 1. Today we'll be looking at population genetics and nature selection. These are two of our substrands. First of all, lesson objectives. We'll be defining these terms: gene pool, gene flow, and genetic drift. Then we'll be listing the conditions for Hardy-Weinberg's equilibrium and how to use his formula and calculate the number of genes present in a gene pool. Let's look at some of these definitions first. First one is gene pool. Of course, it is the sum or total of all the genes, genetic information of various alleles present in a population, or all the alleles of a loci in all the individuals in a population. Gene flow, movement of genes from one population to another, or from one part of the population to another. Genetic drift, random fluctuations or changes in gene frequencies which occur in small populations as a result of chance. These are three definitions that you need to know in order to understand the Hardy-Weinberg's equilibrium or Hardy-Weinberg's principle. Now, Hardy-Weinberg's principle, these were the two different scientists. Okay, one was a mathematician and the other one was a physician who came up with this equilibrium, which says that if mutation do not occur, and if the population is large, and if migration does not occur, then allele and genotype frequencies in a population remain constant, generation after generation. So here, their conditions were, if a population has to maintain its equilibrium, then it has to have all these conditions in it. That means no mutation, no natural selection, there should be random mating happening. Okay, population should be very large. Okay, the small population will always create, will always try to break the equilibrium. There shouldn't be any migration or immigration. No one moving out, no one coming in, and no genetic drift. Genetic drift, okay, that means random fluctuation not allowed. So these were the conditions that Hardy-Weinberg's equilibrium stated. Okay, so this is what they said. Okay, it was hypothetical, non-evolving population, okay, which preserves allele frequencies. Natural populations were very rare. Okay, the useful model to measure if forces are acting on it, and they used this equilibrium to measure the evolutionary change as well. And this is their equilibrium, the principle. Okay, homozygous dominant and recessive genotypes. Okay. In our notes, we have looked at different types of examples of the Hardy-Weinberg's equilibrium. You can go over it and you can redo those exercises again to understand how to calculate our homozygous, heterozygous, and homozygous recessive and homozygous dominance. Natural selection. Okay, natural selection, of course, a theory that was proposed by Charles Darwin and Alfred Wallace. Okay, adapted to the environment, which tend to survive and multiply those that are less well adapted. With the nature selection, there are three conditions that were related to it. First one is stabilizing selection. This type of selection, it maintains the population in a stable form. That means it is going to favor both the phenotypes over the extreme phenotype. So that's how the stabilizing selection graph will look like. Next one is directional selection. Favors the phenotypes of one extreme over the average phenotype and over the phenotype of the other extreme. So that means original population will shift from one side to another extreme. Okay, because that extreme is favoring them a lot. So directional selection. Next one is disruptive selection. That means some kind of disruption will be there for the population to have two extreme phenotypes, okay, over the average phenotype. Therefore, they will have a fluctuating graph. Okay. Now with those selections, speciation will occur. Speciations, two different types of speciation, of course. Yes, we know what is speciation is formation of new species. And how will that happen? Through allopatric speciation and sympatric speciation. Allopatric speciation, there will be a natural barrier coming in, okay, natural barrier. And in sympatric speciation, that means population will be selected for to live in an environment in the given habitat. Okay, and what hinders them is the reproductive barriers. Okay, there are two different types of reproductive barriers, prezygotic and postzygotic. Under prezygotic, you have four different types, which is temporal. That means two species mate at different times of the year. Then we have ecological. Two species inhabit similar regions but occupy different habitats. We have behavioral. Species that respond to specific courtship pattern. Okay, examples are given there as well. And mechanical isolation occurs when genital differences prevent copulation when flowers are pollinated by different animals. Post-zygotic isolation, we have hybrid inviability. Hybrids are produced but fail to develop to reproduce maturity. Then we have hybrid infertility. Okay, hybrids fail to produce functional gametes. For example, if a mule is going to mate with a male donkey, of course, when a male donkey is going to mate with a female horse, a male will be produced, which will be a sterile hybrid. Then we have hybrid breakdowns. Okay. That means fertilization of the F2 generation will fail to develop or are infertile. And the examples are given there. There are other examples that are given in your notes as well. You can refer to your notes for further breakdown of your knowledge. Right, that's the end of this session. In the next lesson, we'll be looking at last part of strand 1, which is evolution.